Welcome to this explainer. Today we're diving into a genuinely high-stakes medical mystery. We're going to investigate the hidden engine of cancer recurrence, a really stealthy mechanism that has frustrated doctors and, sadly, devastated families for decades. But more importantly, we're going to look at a microscopic hero. It's a molecular intervention that recent peer-reviewed oncology research suggests might actually have the power to dismantle this unseen enemy. Okay, let's dive right into this.
We're actually skipping our standard agenda today to plunge directly into a pretty devastating question. Why does cancer come back? You know the story. A patient goes through completely grueling treatments, the scans come back clear, the doctor says the magic word, remission, but then months or even years later, the cancer returns. And when it does, it's almost always more aggressive and way harder to treat. To understand the solution here, we first have to fundamentally rethink our entire understanding of the tumor itself.
Take a look at this quote from the research. It perfectly outlines the core problem. Malignant tumors aren't just uniform masses of bad cells. They actually operate almost like abnormal organs. And hidden deep inside them is a tiny subset of cells known as cancer stem cells, or CSCs. So while our standard therapies are incredibly good at shrinking the main bulk of the tumor, the survival of these unique cancer stem cells is the real primary engine driving that tumor's recurrence.
Section 1. The armor. Why conventional therapies fail.
So, why do our absolute best weapons fail to eliminate this specific type of cell? Well, think of conventional chemotherapy and radiation, sort of like heat-seeking missiles. They're explicitly designed to target and destroy cells that are actively and rapidly multiplying. Those are the fast-moving bulk cells. But cancer stem cells are totally different. They are the silent, deep-running stealth submarines. They can just remain in a quiescent or dormant state. And because they aren't dividing rapidly, the chemotherapy drugs and the radiation simply miss them. Not only that, but these cancer stem cells possess highly advanced defense systems. They literally have mechanisms to repair their own DNA after radiation damage, and they have these specialized pump proteins that actively push chemotherapy drugs out of the cell before they can do any harm. It's wild.
Let's walk through exactly how this terrifying process unfolds clinically. Step 1. The bulk tumor is destroyed by conventional treatment. Step 2. The patient appears to be in remission, but in Step 3, the surviving cancer stem cells do what they do best. They self-renew. A single surviving CSC can divide and differentiate into regular, fast-growing bulk cancer cells. And this leads right into Step 4. They spawn an entirely new generation of tumor cells, causing a complete recurrence. But it actually gets worse. The new tumor inherits the highly resistant traits of the stem cell that birthed it. This is exactly why recurrent cancer is so aggressive and often totally resistant to those initial treatments.
Section 2. The ultimate infiltrator. Enter molecular hydrogen.
So how on earth do we target the seemingly untargetable? Enter molecular hydrogen, or H2. It's the lightest, most ubiquitous gas in nature. What makes it so incredibly special in a medical context is its elemental simplicity. Just to put its size into perspective for a second, it is a fraction of the size of a single oxygen atom. This means it acts as a cellular special ops agent. Many supplements and drugs require specific transport systems or receptors just to get into human cells. Hydrogen? It doesn't need any of that. Because of its minimal molecular weight and size, it easily diffuses through the walls of the alveoli and the lungs, goes straight into the blood plasma, and slips right past those advanced biological defense systems we talked about. It penetrates biological barriers directly into the cytosol, the mitochondria, and the nucleus of the cell.
1 minute. That is the crucial point here. Speed and bioavailability, meaning how quickly the body can actually use it. The research notes that with intravenous administration of isotonic solutions saturated with hydrogen, the peak breath concentration of the molecule is reached in just 1 minute, literally 60 seconds. Even through simple inhalation, it rapidly diffuses into the tissues, overcoming those barriers to initiate cellular effects almost immediately.
Section 3. The takedown. Defeating cancer stem cells.
Okay, so now that we know hydrogen can physically get inside the cell, let's look at what it actually does when it encounters our antagonist. The primary scientific proof of hydrogen's power comes from its ability to disrupt the cancer stem cell's biggest superpower, self-renewal. In the lab, cancer stem cells form these microspheres, which are essentially little clonogenic clusters where they multiply. But when researchers treated human ovarian cancer cell lines with a hydrogen-rich medium, they saw that molecular hydrogen markedly inhibited their sphere-forming ability. In fact, in one model, hydrogen treatment led to an 82% reduction in the number of spheres. By cutting off this self-renewal capacity, hydrogen prevents the cancer stem cells from dividing, effectively forcing them to differentiate. And that's huge. It means they turn back into regular, manageable cells and completely lose those stem-like, resistant qualities.
Notice the cell assays right here. The data proves that hydrogen doesn't just stop them from cloning themselves, it practically paralyzes them. The research highlights that hydrogen treatment significantly inhibits cancer cell proliferation, invasion, and migration. It literally stops these cells from moving into and invading surrounding healthy tissues, essentially locking them in place.
Section 4. Evidence from the battlefield.
Now you might be wondering, does this microscopic battle hold up outside of a petri dish? Let's take a look at the in vivo animal models. Researchers took live animal models, specifically mice grafted with human ovarian cancer tumors, and placed them in a transparent chamber. For 2 hours a day over a span of 6 weeks, these mice inhaled an atmosphere containing a 67% molecular hydrogen mixture. And the visual readouts? Highly significant. Tumor growth was visibly and measurably suppressed compared to the control group.
Let's break down those massive battlefield victories by the numbers, because they are stunning. After 6 weeks of hydrogen inhalation, the mean tumor volume dropped by 32.3%. But the biomarkers reveal exactly how this happened. The expression of Ki-67, which is a major marker of cell proliferation, went down by 30%. Even more staggering, the expression of CD34 absolutely plummeted by 74%. Now, CD34 is a marker for angiogenesis. That's the process by which a tumor grows brand new blood vessels just to feed itself. By suppressing CD34 by 74%, molecular hydrogen essentially chokes the tumor. It completely cuts off the blood supply that feeds the developing mass and its stem cell niches.
Section 5. The human element.
So we've seen it in cells, and we've seen it in mice. But what about us? What about humans? A study actually followed 82 patients with advanced stage 3 and stage 4 cancers who had already failed conventional treatments. They inhaled a hydrogen-oxygen mixture for at least 3 hours a day. After just 2 to 4 weeks, the improvements in their daily quality of life were absolutely profound. The data shows significant reductions in fatigue, nausea, and insomnia, right alongside really notable improvements in appetite, cognitive function, and pain management. Overall, 41.5% of these advanced stage patients experienced a highly quantifiable improvement in their physical status.
But listen, it wasn't just about feeling better. The clinical victories here were staggering. Out of 80 cases with tumors visible on imaging, the total disease control rate was 57.5%. When you isolate just the stage 3 patients, that disease control rate skyrockets to 83%. On top of that, of the patients who started the trial with elevated tumor markers, 36.2% saw those markers actually decrease. And that initial decline happened in a median time of just 23 days. The researchers noted that lung cancer patients in particular saw some of the most dramatic physical improvements and tumor marker decreases. And, crucially, this is huge, there was no hematological toxicity observed, meaning it didn't destroy their healthy blood cells the way traditional chemo does.
Section 6. A new horizon.
Well, we've covered a lot of ground today, looking at the very mechanics of how cancer survives and how it might actually be stopped. The treatment of advanced cancer really is one of humanity's greatest challenges. Conventional treatments, while absolutely necessary, often leave behind the very cancer stem cells that guarantee a relapse, sometimes even promoting the very inflammation and resistance they're trying to cure. But here we have molecular hydrogen. It's a simple, low-cost, low-toxicity therapy that effortlessly permeates biological barriers to choke off tumor blood supplies, freeze cell migration, and force those incredibly resilient cancer stem cells to stop cloning themselves. It really leaves us with this final provocative thought to take away today. Could the universe's absolute simplest molecule actually be the key to defeating our most complex disease? Thank you so much for joining me for this explainer. Keep questioning and keep learning.